Hi students, let's continue with food hazards and overview. We have seen different sessions, what are the hazards and what are the different results of the presence of such hazards and what would result into the qualitative deterioration of food. Now today we're going to talk about the microbiological hazards and how they have to be detected within our food. We have already discussed the microbiological hazards, but today's session will mainly help you to detect the pathogens in the food. Microbiological hazards or the microbes are usually isolated by conventional methods, and usually we have the culture and the colony counting method, which is the most primitive method. Previously, that was the only method which was being used. That is, you culture the organism onto a culture plate and then you identify the organism. Further on, different other methods like using immunological techniques as well as polymerase chain reaction based techniques have also been advanced right now. Coming to the culture and the colony based methods. Here, standard microbiological techniques are mainly used to detect the single bacteria. The bacteria are grown or cultured and they are being identified, and further on subsequent diagnosis is being done. By appropriate procedures, they have to be grown separately on culture media and obtained as a pure culture for the study. You can use different media, like selective media can be used. Selective media is a substance in which some inhibitory substance is added into the solid media so that the specific organism which has to be identified would be preferred to be grown, whereas the others would be inhibited to grow. Now, in the case of gram-negative bacteria, you can use McConkey's agar. And if you want to isolate Vibrio cholera or Vibrio species, you can use thiosulfate citrate bile salt sucrose agar, or called the TCBS agar. I repeat, thiosulfate citrate bile salt sucrose agar for the isolation for Vibrio cholera. And we know for E. coli detection, we use the EMB agar or the EOC methylene blue agar. For the detection of Mycobacterium tuberculosis, we have the LJ media or the Lowenstein-Jensen media, which is specific for MTB or Mycobacterium. Even various other species of Mycobacterium also can grow on LJ media. So, if you want to isolate that particular organism, you have to take a food sample or whatever it is, and you have to check whether it is capable of growing. If an organism grows on LJ media with a characteristic colony of Mycobacterium, you can identify that probably that particular organism is there in the medium. In the case of Salmonella typhi, we can use Wilson and Blair medium. And in the case of diphtheria bacilli, you can use potassium tellurite media. Now, something which we have to know is that these are not the only specific media which are available. Every organism can have different types of selective media. So based upon the availability of what selective media is there in the media, as well as what is being considered to be as a standard media, you can use different media for the same organism. Now this is a picture depicting. Now you can have here the McConkey agar to show the lactose fermenting colonies. Usually the lactose fermenting colonies will appear to be pink in color, whereas the non-lactose fermenting will appear to be whitish in color, whitish or creamish in color. Now in TCBS agar, this is TCBS agar which we are showing here. It is a selective media for what? For Vibrio cholerae. If Vibrio cholera comes, it will get a yellowish color, and other species of Vibrio will be having a different color. Now, in the case of EMB, it will appear to be as if E. coli grows, it will appear to have a greenish, blackish metal with a greenish, it will appear to be with a greenish metallic sheen or a blackish metallic sheen, greenish black metallic sheen for the E. coli colonies. The others will lack this, and thus you will be able to identify whether it is a E. coli strain. Coming to the immunological based methods, one technique that we have is ELISA, or the enzyme linked immunosorbent assay. Here you are using the presence of antibodies. The antibody antigen reactions are used for the detection of the particular pathogen. This technique can be used for various pathogens like Escherichia coli, Salmonella, Listeria monocytogenes, Staphylococcus, etc. Different types. You should note that Listeria monocytogenes, M should be written as a small letters. I have made an error in my slide. Just kindly hope so you don't make that mistake, because always the species name has to be written in small letters. So anything which has to be detected, the you can use that one's antigen antibody based reaction to detect whether the antibody is being present here. Firstly, what happens now? Here, what you should have a basic idea. First of all, you remember that against every antigen, your body produces an antibody, and that antibody is being used here for the ELISA based technique. Now, I'm not telling, you might think, how come an antibody is being formed and how will you analyze that in a food? So basically, to analyze if a particular, what you can do is you can only check whether the antigen is being present in the food. So, synthetically or using various other techniques like monoclonal antibodies, so that is by using different mouse based techniques, the antibodies which are being produced against each antigen is being produced in a lab separately, and you will get antibody coated wells. Now just look at this well. This is a well. It is usually done in small plates called the microtiter plates. You can purchase these microtiter plates which are already been coated with antibodies, or you can coat them with an antibody. So you these antibodies are mainly being produced in a separate lab against different pathogens, against probably E. coli, for example. And then you're taking the plate and you're coating it with an antibody. After that, the proteins, okay, proteins here, what does it means? It is an antigen. Okay, you take your food and you homogenize it and you process it, and in that probably what will be there, the antigens of Escherichia coli. Antigen is nothing but on the surface of E. coli there would be certain markers. Just now if you consider yourself as an antigen, consider yourself as an antigen, on your body there would be certain molds or birthmarks indicating which is probably that you are that person. Similarly, on the surface of Escherichia coli, there would be certain proteins protruding from its on its surface, which would indicate that it is Escherichia coli itself. I'm not going to the more details of that. They can be referred to as the antigens. Now, if E. coli is there in that particular food sample, that particular proteins or the antigens, here these green, yellow and all that. Now here, if it is E. coli, it would contain this particular green color protein. And so when you mix a homogenate of the a sample of the food homogenized food product, if E. coli is being present, that will come and bind to the E. coli specific antibody. So what happens? Then you will excess of that which is being present here will be washed. If E. coli is being present, it comes and binds to the E. coli specific antibody, and after that a second antibody is being added, okay, which contains an enzyme linked to it. That is called the secondary antibody. That is only an antibody which is also specific to what? This E. coli antigen will be provided in the kit itself. In the ELISA kit itself, it will be provided. And that would be linked with some enzyme. Okay, so the secondary antibody binds to the protein, and then what happens? A visual change indicates the presence of there would be a colorimetric reaction which will get changed by the action of an enzyme will be acted upon this antibody, and a substrate. This antibody is also being linked with an enzyme. So that enzyme will further act on a substrate. So and convert the substrate into a colored component, and that could be detected. So what you are, I'll repeat this process of ELISA once more. If you have to detect a particular antigen, means the presence of any bacteria, the antibodies of that are specifically bound to a titer plate. This antibody is not being synthesized right now in your body. In the lab kit itself, it will be provided. So you take the titer plate. Sometimes the titer plate itself will be bound with the antibody, or you will have a specific reaction to bind the antibody onto the titer plate. After that, the food sample which has to be analyzed is homogenized, and you expect that if there is the bacteria present in that, there would be a specific antigen bound to the bacteria, and that will come and bind to this antibody. This antibody is in such a way that it will bind to only one type of antigens. The remaining excess of the food, which is of the proteins which are here, are being washed away, and further on a secondary antibody is being bound. If and this secondary antibody is also specific to that particular antigen. Now if this green color is E. coli this antibody would also be specific to it, so it comes and binds to it. In some cases, it would be specific. Yeah, it's specific here. And what happens? There would be an enzyme linked to it. Okay, and once the enzyme is being bound here, you will provide a substrate. Okay, and this enzyme will convert the substrate into a colored component. If and you should remember, after each and every step, now once you add this here, you will just wash it. So if there is any unbound secondary antibody, that will just wash out. And the secondary antibody will be bound here only if this green color antigen is being present here. So only if the secondary antibody is present, a colored component comes. And only if the antigen is present here, the secondary antibody can bind here. So in turn, by determining the color of the product which is being found by the action of the enzyme linked to the secondary body, you are finding out whether the antigen is being present. I hope this is clear to you right now. So this is the process of a sandwich ELISA, which is being used for the detection of food samples. We will be talking about the PCR based detection in the next sessions. Now the antigens the antibodies, like the same process can be used for different bacteria. In different bacteria, the different antibodies which have been bound here would be different. Thank you for now, dear students. We will continue in the next session, the PCR based analysis of food pathogens. Thank you once again.